Hello, I'm Marianna. I'm a third year PhD student studying the regulated interaction between 1433s and the protein kinase named ALK1. This project is shared between the laboratories of Professor Carol McIntosh and Dr. Ian Galley at the University of Dundee. My project is part of a European training network called TASPI, which stands for Targeted Stabilization of Protein-Protein Interaction, which is in collaboration between 6 European universities and 5 industry partners. Our work is centered on reversible protein phosphorylation, which is catalyzed by protein kinases and protein phosphatases. This is a molecular switch that is responsible for controlling almost every event inside our cells. Fisher and Krebs won a Nobel Prize for their discovery of this vital mechanism. Importantly, this regulation of these protein phosphorylation switches promotes many human conditions, including cancer, diabetes, and neurological disorders. Over the past 30 years, there has been exciting progresses in developing drugs that inhibit specific protein kinases to treat diseases. However, TASPI is taking a different approach to drug discovery in the protein phosphorylation field. The TASPI approach is based on the fact that when certain target proteins are phosphorylated on specific pairs of residues, these phosphosides then dock onto C-shaped proteins that are called 1433s. The chemists in TASPI are developing small molecules that act as specific molecular glues that fit in here and stabilize the binding of the 1433s to the specific target phosphoproteins that are affected in various diseases. This means that the drug alters the function of the target phosphoproteins so that the disease might be corrected. The specific target phosphoproteins that I am studying in my PhD project is called ALK1. ALK1 is a protein kinase responsible for autophagy initiation. Autophagy is a series of steps whereby membranes assemble into these autophagism structures that engulfs cellular content for digestion and recycling. Autophagy dysfunction is involved in lysosomal storage disorders, cancer, and neurological diseases. Autophagy is triggered when ALK1 is activated by a pathway that senses when cells' nutrients and energy levels are low. So I was fascinated to find that ALK1 has one 1433 binding phosphoside that is phosphorylated by the same energy sensing kinase AMPK. In contrast, the second phosphoside on ALK1 is phosphorylated in response to insulin and growth factors. And both sides promotes 1433 binding, which then presumably activate ALK1 and triggers autophagy. So now I want to know more about regulation of 1433 binding to ALK1 and how 1433 binding to ALK1 affects autophagy. And with my TASPI colleagues, I will test how autophagy is affected by small molecules that glue 1433 onto ALK1. Finally, I should mention that in Dundee, I've had the opportunity to use a variety of advanced techniques in my studies. For example, isolation of GFP-tagged proteins from transfected cells and using phosphoproteomics to pinpoint specific phosphosites on ALK1, and specialized visualization techniques to identify which cellular stimuli promote the phosphorylation of ALK1 and binding of 1433s. Thank you very much for your attention. I'm happy to take any question or feel free to email me at any time.